My name is Suzie Chen. I'm a professor in the Department of Chemical Biology, School of Pharmacy at Rutgers. The main focus of my research is on the molecular mechanism of melanoma development. My goal is to understand how the disease comes about. If you understand the basis of how the diseases evolve, how they change, and perhaps you can design better ways of combat. So we have used mouse models to, as a model system to investigate using cell lines in culture, using animals as the next step once we discover something in culture cells to see how it reflects the in vivo situation. And then hopefully we will translate the finding to the human system. And I do a lot of work with investigators over the Cancer Institute in New Jersey. We actually have translated a few findings into the human system where we have several clinical trials that's based on our preclinical work here in my laboratory. The main thing that we try to discover is really based on work that my lab has uncovered, where we identify a normal neuronal receptor that deals with learning and memory. When it's inappropriately expressed in melanocytes, so melanocytes are the cells that's in your skin that gives you the pigmentation in the skin. So normally these cells, they do not express this receptor. So when the expression of the receptor is turned on, a consequence is tumor development in culture cells. So this receptor normally is expressed in the neuronal cells. It actually, if you don't have this receptor, or if you genetically sort of delete this receptor or make it inactive, a consequence in the neural is deficiency in learning and memory. So that's only in the neural. But when it's inappropriately expressed in melanocytes, the skin cells, the consequences of tumor development. So it kind of shows you the expression of gene in different cell type actually have different consequences. We went back to the cell lines and asked, do you see this type of expression not only in the mouse system but also in human melanoma cells? And we found that that's indeed the case. From the human cell lines, we went on to look at human biopsy samples and stuff that's taken from the patient that has entered the Cancer Institute in New Jersey. We also saw the expression of the receptor in several biopsies that we knew that not only is expressed in the mouse as a model system, we actually found the human relevance in not only the human cell lines but also the biopsy samples that we looked at it. My main thing is to investigate, how does this actually occur? How does expression of a receptor, it gets turned on? It's difficult to do because, you want to go back to the human and ask, well, how does it turn on in the human? I know in the mouse, well, I kind of made it that way. But to really go back and forth between human and mouse, sometimes one thing you see in the mouse system, they don't always translate into the human system. So it's a little bit tough, and plus work you do in culture cells, it doesn't mean always going to reflect in the, say, a real human. Because when you grow cells in culture, a lot of things will change. So perhaps expression of this receptor is one of those consequences. But that's why it was important for us to look at biopsy samples. So the conclusion that we draw, again, is that what we observe in our model system, in the mouse system, actually we prove that it actually happens in the human system. So a lot of things you learn from the model system, then you want to go back to ask, does this happens in the human or not? Because after all, human health is really the major goal, of all of us doing basic research. The application of my work, melanoma, believe it or not, it's a very, very aggressive disease. If you discover early, you go to a dermatologist and surgeon, they take it out, you can be disease-free forever, perhaps, for 10 years, for 20 years, it depends on how quickly you go to your dermatologist, how early you have that mole or whatever removed. If you just ignore that, it will then spread to other places. Once that happens, the chance of you living beyond 5 years is very, very small. Even though you have the primary tumor removed early on, it may come back. When it comes back, it becomes a very, very aggressive disease also. it kills people. So there's still really a great need to try to find so-called a cure for melanoma, because we're just not quite there yet. You know, cancer cells are very small. You hit them with one thing, they turn around and they change, and you have a totally different thing to look at it. So we will continue to understand how this receptor, you know, just by its expression can elucidate this entire, you know, pathway or become a tumor, and become a tumor that's very, very metastatic. So we try to find, using our model system, to understand it, and then go back to the human and ask, does this really also apply as well? So as I said before, it's kind of back and forth. You go from the laboratory to the clinic and back, and just by using the knowledge that you gain to go a small incremental step all the time. I have had many graduate students working under my supervision, from various programs for the molecular biosciences to the toxicology, pharmacology graduate program. And I'll have to say, each of my graduate students, when they finish with me, they all have secured fairly good jobs. Some of them are in academia, but most of them are in industry. And I also have had undergraduate students or even high school students that has come through my lab, many of them over the years, either during the school year or in the summer. But through School of Pharmacy, there is a summer undergraduate fellowship that undergraduate students from not only Rutgers, but really any place that could apply. And they will spend something like 10 weeks in the laboratory performing work. As a basic scientist, it's gratifying for me to know that my work actually perhaps has made a very small contribution to human health, but it doesn't happen all the time. So I consider myself pretty lucky in that aspect.